right, so I'm just going to recap what we already did on Thursday, so I'm not going to stay on any of these slides for a super long time. On Thursday, we started talking about cell respiration, and I called it Destiny because I asked you what was happening to glucose. We added in some definitions here about aerobic and anaerobic cell respiration and about fermentation and when those things might happen. The cellular respiration overall reaction is the one we wrote here. So we're talking about 1 glucose, 6 oxygens, making 6 CO2, 6 water, and 36 ATP if we are lucky. I also reminded us all that we still need to know these two parts of the mitochondrion, specifically the matrix and the cristae, because that's where 2 of the parts are going to happen. Then we talked about what happens when glucose comes into your body. And then I mentioned something that I'll just highlight. Your liver is so super duper important. It plays a role in a whole bunch of different body systems. And I said that if I were to ask a written question on the final, this would make an excellent choice because it has to do with all of your body systems. And then we talked about where does glucose go? Where does it end up depending on whether we're using it or not? This piece over here is saying that we are going to do cell respiration. That would be what we do with glucose if we need it. If we don't need the glucose, there are 2 ways that it can be stored. Now, glycogen happens in your liver and in your muscles. Fat, of course, happens in fat cells. And excretion is where we mentioned kidneys specifically, and I alluded to the fact that we would be dissecting kidneys later when we talk about that chapter. So that's what we had done before. And then I gave you this overview, and this is sort of where we ended. So we're talking about glucose oxidation. And I'm going to highlight that word once again. To take CO2 and make glucose, I had to reduce it. Now I'm going to do the opposite. I'm going to take glucose and oxidize it, and I'm going to get back CO2. Now, glycolysis is the first step, and I drew it outside of the box because the box represents a mitochondrion. So this is explaining that the first step of glucose oxidation does not have to happen in a mitochondrion. And neither does fermentation if that's the path that we're choosing. The other 2 parts of full cellular respiration happen inside of a mitochondrion. So what we're saying is prokaryotic cells can do cell respiration. They just can't do all of the steps. Eukaryotic cells, so anything that has a mitochondrion, all eukaryotic cells can do those 2 parts. Any cell, prokaryotic or eukaryotic, can do the first part. They don't have to have a mitochondrion. So we're going to talk about each of the steps that happen sort of one at a time. After the first step, there's kind of a choice here. And that's the distinguishing factor. Is there oxygen or is there not oxygen? And that's how we'll make the decision after the first step of cellular respiration. So now on to things that are new. Glucose oxidation step number 1 is called glycolysis. If I look at the word, it has lysis in it, and this is the 3rd time that we've seen that piece of a word. Glycolysis. We've seen photolysis. We've seen hydrolysis. Lysis means that I am breaking something apart, splitting something in half. And glyco is talking about my sugar molecule, in this case, glucose. So what do I know about glycolysis? First of all, it occurs in the cytoplasm. So that means I do not have to be inside of a mitochondria for this step to happen. This step does not use oxygen. So organisms that are living in anoxic conditions, or organisms that do not have oxygen available to them at that moment in time, can still do this first step. Question? Nope. Okay. Now, what is the overall point of this step? That's this third thing. I'm going to take one glucose, which has 6 carbons, into 2 pyruvic acids, which each have 3 carbons. Now, at this particular step, I'm going to point something out just about chemical nomenclature. The way that you name acids is always based on what the substance is before I put it in water. And so you will see at different points the substance produced at the end of this referred to as pyruvate or pyruvic acid. The only difference is that for something to be an acid, it has to be in solution. And pyruvate is the substance that would get dissolved in water. That's a chemistry point. I'm not asking you to be able to explain that. I'm just letting you know that you might see the word pyruvate here, and I'm talking about the same substance, pyruvic acid. So that's the point of this. I'm taking glucose, I'm splitting it in half. What do I get from glycolysis? What are the products? I get 2 of these pyruvic acids, or pyruvates, 2 NADH, and I technically get 4 ATPs, but I'm putting a star there. Now, the reason I put a star is that even though it's true, I am making 4 molecules of ATP, I also have to use 2 ATP to get things going. That means that even though I'm making 4, since I had to use 2, the net amount, the overall amount of ATP that I'll have more than when I started this process is only 2. Now, the reason we have to use 2 ATP is to do something called phosphorylate glucose. Essentially, I'm adding a phosphate group to glucose. The reason we would do this is to prevent it from diffusing out of the cell so that it doesn't go away, because glucose can diffuse in and out of the cell. It also makes it more reactive. So those are the basic things that you'd need to know about glycolysis. It happens in the cytoplasm. It does not use oxygen. Its goal is to split my one glucose into 2 pieces. Glucose has 6 carbons. Each of the pieces has 3. When I do that, I produce those 2 pyruvic acids, 2 NADHs, and 4 ATPs, although because I had to use 2, I only have 2 net ATPs. Now, over on the right is a picture representing the steps that are happening in glycolysis. And I'm going to highlight the pieces that you need to really be aware of. You absolutely have to know that glucose is the starting point. Now, the next compound, fructose diphosphate, you do not have to know that particular name. I'm never going to ask you that particular name. I'm just going to highlight that these 2 things both have 6 carbons. And after that point, I split that apart into things that have 3 carbons. Now, does it interest me that you know the names of all of the intermediates? No, and some of them aren't even listed. The only names I need you to know for the carbon compounds are the beginning and the end. Glucose goes in, it has 6 carbons. Pyruvic acid comes out, it has 3 carbons. Now, in terms of what's happening or what I'm getting or what I'm using, at the top, I am taking 2 ATP and they're losing their phosphate group. So it's mentioned over here on the side, but I'm just going to write the word again. This is phosphorylation. I'm using ATP, I'm taking the phosphates, they're adding onto glucose. I need them so glucose doesn't leave, so it continues on with this process. Then I have a whole bunch of reduction happening. I had a 6-carbon compound and I split it in half. So that means that this next set of reactions happens 2 times because there are 2 of these molecules. NAD, now notice, it's not NADP. NADP positive is for photosynthesis. So I always remember the one with the P is for photosynthesis. The one without the P is for cell respiration. So that molecule is getting reduced. It happens on both sides. ATP is getting made, so I'm reducing ADP. I'm going to write the word chemiosmosis here because that's how I get ATP all the time. And then the last thing that I'm going to add is this. If NAD is getting reduced, this carbon compound is getting oxidized. So remember, oxidation and reduction both happen at the same time. In cellular respiration, the thing with carbon in it is always the thing getting oxidized. The other molecule is generally the thing that is being reduced. Now, I would not ask you to draw this thing out from scratch because we've left out a bunch of intermediate names as well. I would expect you to know what happens during the overall process. So could you, for example, tell me what the overall products are? That's something that's very important that you know. It's also incredibly important that you can understand that I'm getting 2 net ATPs. I am producing 4, 2 over here, and 2 over here, but since I had to use up 2, I subtract them from my overall total. Now, would anyone like to ask any questions about this first step of glucose oxidation? So this is step number 1. What we'll talk about next is where does that pyruvic acid go after glycolysis? Because there's more than one choice, and it really depends on does the organism have oxygen available or not, and or does it have a mitochondrion? That's really the distinction that we're looking for. So, the first possibility for what happens to pyruvic acid is anaerobic respiration. Now, anaerobic respiration, I'm grouping together with fermentation. If you recall the other day, fermentation was its own category. They're both based on no oxygen available. The difference is that anaerobic respiration happens in organisms usually who would perform cell respiration aerobically, but they can't. Fermentation happens in organisms that normally wouldn't produce or perform cell respiration with oxygen. Now, when does this happen? Well, in us, it happens in your muscle cells. And the time that it would happen would be, for example, during really strenuous exercise. The idea is that your cell is using oxygen faster than the circulatory system can provide it. So if you have ever felt the burn, this is what's happening. This is the burn. When you're feeling the burn, the reason you're feeling it is because of anaerobic respiration. Now, this is what happens in us, humans, and generally in any sort of animal, this would happen. It can also happen in other organisms, like a yeast cell. When it happens in yeast cells, the product is alcohol. So a lot of you are aware that to make different alcoholic beverages, which none of you have tried because you are not old enough, you don't even know what they look like or smell like, but you just might be aware that they exist, alcohol is made from fermentation. That's how you produce it. And depending on which type of plant we are fermenting, you get different types of alcohol. So this is yeast. We are actually going to make yeast accomplish cellular respiration in our class tomorrow. So that will be exciting for you. It will not make alcohol of any valuable content, just so you know. It would not be tasty to consume it. There we go. So what's going to happen during anaerobic respiration is that we're going to take glucose and we're going to make one of two things. Lactic acid or alcohol. Now we're really going to focus on the lactic acid part because that's what happens in you. And our next chapter is muscles, and so this is a very nice lead-in into what's happening there. Now here's an interesting point. Not all of your cells can do this. For example, your brain dies without oxygen. Your brain cannot function without oxygen. Side note, your brain can't function without glucose either, so please do not do a no-carb ever diet. You will die. Your brain cannot function without it. But not all of your body cells can do this. So if I were to ask you on a test, which kind of cell does this happen in? A heart cell, a muscle cell, a kidney cell, a brain cell? You would say anything but brain. Brain is the only type of cell that I would expect you to be aware of that cannot do anaerobic respiration. Now, side note, because this is my area of research. When I was in university, this is what my degree was about, brain stuff. And one of the things I studied were strokes. And the fancy word for a stroke is cerebral ischemia. And if anyone were ever to ask you, how did that person die, that is the answer. The thing that kills you, regardless of what the immediate cause was, is that your brain doesn't have oxygen. For example, if you get shot and you bleed out, you die because your brain doesn't get oxygen. If you drown, you can't breathe, you die because your brain doesn't have oxygen. If you get your head chopped off, your brain doesn't get oxygen, you die. If there was a way to keep oxygenating your brain, it could stay living, which is what a lot of sci-fi things are based on, a head in a jar on a robot's body. That's realistic in the sense that your brain requires oxygen to survive. If you could keep providing that, then you could keep living. Now, would it be a quality life? Probably not if your head is no longer attached to your body. But I'm going to write the word. This is the word, ischemia. It's what has killed everybody ever that has ever died, ever, this thing. Now the next point, and the next few points are about what lactic acid does and how your body overcomes this. So lactic acid is an acid. It decreases pH. Acids have a low pH. The result of this is that your muscles cannot contract. So your muscles cannot perform the full contract-relax cycle. And actually, I'm going to add this in. They can't contract and then relax. And so if you do not have enough oxygen, you do not have enough energy for your muscles to contract and then let go again. And this contraction that's not letting go is probably what's making your muscle hurt. Now, how do you overcome this? You have to meet the oxygen debt. So being in debt means I don't have enough. When I meet the oxygen debt by breathing and heart rate increasing. So I have to breathe faster, my heart has to beat faster. That increases the supply of oxygen to muscle cells, and so that stops this process from happening. Now, lactic acid is not useless. Your body doesn't do things that are useless. Lactic acid can be converted to glycogen, which we know is a storage form of glucose, and I still get 2 ATP from this process. So I'm not getting nothing, I'm just not getting as much as I otherwise would have. So over on the right is a little diagram trying to illustrate how I go from glucose to pyruvic acid to alcohol or lactic acid fermentation. So if you look, this part right here up to the green line, that's all glycolysis. That's the part that we would have already discussed. This sums up that I have to use 2 ATP, and then I produce 4 ATP. It also reminds you that glucose is getting oxidized, so something else must be getting reduced. Now, once I have that pyruvic acid or pyruvate, I have two choices: yeast fermentation or homolactic fermentation. Homolactic, homo means Homo sapiens, people, muscles. Lactic is referring to lactic acid. Since all of this is happening in the cytoplasm, which is sometimes referred to as cytosol, it doesn't matter what kind of cell you are, any cell could do one of these things. Now, does anyone have any questions they'd like to ask about anaerobic respiration? What happens when there is no oxygen available? So before, I'll just repeat this because everyone is coming back. Before you write everything down, maybe take a look and notice this was all on the last page. That's anaerobic respiration. And I just wanted to show you how it was connected to aerobic respiration. Here's my pyruvic acid right here. If there was no oxygen available and or there was no mitochondria available, my only choice was fermentation. So this side happens when there is no oxygen or no mitochondrion. It could be one or it could be the other. So for example, homolactic fermentation, which happens in your muscles, isn't happening because there's no mitochondria available. It's happening because there's no oxygen. Aerobic respiration absolutely uses oxygen. If oxygen is not available, this part can't happen. Like the diagram illustrates, it occurs in any cell that has mitochondria. And there are 3 stages. The first stage we already know, glycolysis. It happens outside of the mitochondria. It does not require mitochondria to happen, but it has to happen before aerobic respiration can. Now these other 2 parts, I guess I'm calling them A, B, and C lower down. The other 2 parts happen in the mitochondrion. And this is the simplified answer to what do I get from the other 2 parts. I get 34 ATPs, and I get some CO2 and some water. Of course, I'm going to show you exactly how that happens. But aerobic respiration is what happens if I have oxygen and I have a mitochondrion. There's 3 parts: glycolysis, Krebs, and the electron transport chain. So that you know, the electron transport chain is abbreviated ETC, and that's how I refer to it most of the time. And it would be fine with me if you also use that abbreviation when you were discussing it or naming it or mentioning it. And then I mentioned at the bottom, A, or number 1, glycolysis does not happen in the mitochondria, but both B and C do, the Krebs cycle and the electron transport chain. Does anyone want to ask any questions about either anaerobic or aerobic respiration? Maybe what's the difference between them or anything like that? So then we need to talk about how do I get through the 3 stages. Glycolysis we've already mentioned, so we really need to discuss the Krebs cycle and the electron transport chain. Now, the funny thing with the Krebs cycle is that there is actually something that has to happen before it. It's the preparation for the Krebs cycle. And so it often gets grouped in with the Krebs cycle, but I'm splitting it apart so that you can see that it's actually a separate piece, a separate part. And so we could call it, there's 2A and 2B, or there's actually 4 steps, however you want to call it. Most textbooks just refer to these as the 3 official steps, but I like to make sure that you can see how I'm getting from one step to the next. And the Krebs cycle doesn't start with pyruvate. It starts with a different substance, so I have to show you how we got there. So this is where lesson 11 starts. We're going to do a bit of it, finish it off tomorrow. Lesson 11, I'm going to play the song tomorrow. It'll make more sense. Some wonderful people took this Black Eyed Peas song, I've Got a Feelin', but made it be about cell respiration. It's fairly fantastic. But I think we'll save that for tomorrow once we've talked about what goes on in this lesson. So for me to get from glycolysis to the Krebs cycle, there's what we call the link reaction. The link reaction happens in the matrix of the mitochondria. So if I look at my mitochondrion, it's kind of an ovally shaped thing. It's got the wavy line. The matrix is the inner portion. It's the fluid on the inside. So we could compare the matrix of the mitochondria kind of to the stroma of a chloroplast. Now I have this described 2 different ways. The text down here says exactly what the picture up there does. And I put both because I know some people prefer words and some people prefer something more visual. So I'm going to talk about the picture so that you're sure you understand. And the text underneath is exactly what the picture is showing as well. So what do I get from glycolysis? I get something that has 3 carbons. So that's what that number 3 refers to. So that I got my pyruvate, I made that in glycolysis. Another molecule, coenzyme A, shows up. We know what coenzymes are. They help reactions happen faster. These 2 things join up, coenzyme A and pyruvate, and they make a compound called acetyl coenzyme A, which is a ridiculously long name. Some people call it just acetyl-CoA. I've heard it referred to just as CoA. I don't like the one on the top because it ignores the fact that there was some other part to the molecule. But calling it acetyl-CoA is very commonly accepted. And so it would be fine with me if you ever had to write the name of this molecule if you called it acetyl-CoA. Now that's the molecule that goes to the Krebs cycle. How did I get that from pyruvate and coenzyme A? Well, acetyl-CoA has 2 carbons. That's not the same. Pyruvate had 3. So how did that happen? D, which means I undid something, D, carboxylation. I took off a carbon and some oxygen. So see how it says carb, ox? It's telling you what I took away. Decarboxylation. I took off carbon, I took off oxygen. In reality, I'm taking off a molecule of CO2, which is awesome because I knew I was supposed to get CO2 from cell respiration somewhere, and this is one of the places that it happens. Now, what else is happening? Well, oxidation and reduction are both happening. So I'm going to be clear. Oxidation of pyruvate reduction of NAD positive. Because remember, they both have to happen at the same time. I take away hydrogen from my pyruvate. Oxidation is what happens when I'm taking something away. The thing that's getting reduced then is NAD positive. It's becoming NADH. So then we're going to talk about the Krebs cycle, and I think this is the last thing that we're going to talk about new today, and we'll save the rest for tomorrow. I'm going to make this a little bit differently sized so we can see. So we're in glucose oxidation still, step number 2. Now you could call the link, like I said, the preparation for step number 2 or step number 2A. I'm not super concerned with how you number them as long as you know what order they happen. This is titled the Krebs Cycle. We could also call this the citric acid cycle. That's another acceptable name for it. Now, before you try to draw the entire cycle that's on the left, I want to alert you that I am not going to ask you to know the names of all of the compounds that are in that cycle. There are a lot of them. What I am going to ask you to be aware of is what do I get from the cycle. So you'll see that I highlighted a bunch of things in yellow. Those are the things that I'm going to point out. It's all written in words on the side, but since I know some people prefer pictures to words, I put both of them here so that you can see them. The thing you have to know about this cycle, and that's why I'm saying each cycle, is that just like the Calvin cycle in photosynthesis, this cycle happens twice. Just like I needed to do the Calvin cycle twice to make one glucose, I need to do the Krebs cycle twice because every time I break glucose in half and I get a pyruvate, well, I get 2 of them. So everything that's going on there happens twice. So every time the cycle happens once, what do I get? Well, acetyl coenzyme A, which is this little guy right here, comes in. Now, like I mentioned on the previous slide, acetyl coenzyme A has 2 carbons. So you can see there's 2 C's attached to each other. Just like in the Calvin-Benson cycle, there's a molecule ready to accept it. So it says a 4-carbon compound. Well, here it is. It's called oxaloacetate. I won't ever ask you that. I'm just letting you know it has 4 carbons. Acetyl coenzyme A has 2 carbons. They're going to join up and make something that has 6 carbons. It's called citrate. I don't need you to name it. I'm just telling you how many carbons there are so that you can see how I'm eventually going to make the substances that I'm going to make. Now what happens is that this 6-carbon compound, which is technically called citrate, is then broken down over a series of steps all the way back up here to the original 4-carbon compound. See how I'm not focusing on what order everything is happening in here? I'm saying this 4-carbon thing grabs acetyl-CoA, they make something with 6, and then I break that thing back down to the original molecule. So this whole part, all the way around the circle, we're going to group together as what happens during the Krebs cycle. In the process, 2 carbon dioxide molecules are removed. So here's one of them. Here's the other one. Does it matter to me that you know exactly in which order all of these molecules are made? No, I won't ask you that. The other thing that I get is NADH, oh, that was missing a 2, and FADH2. Now, in total, if I look at what I'm getting, I get NADH over here, NADH over here, NADH over here. I get 3 of them. FADH2 only happens once, right there. So I just get 1 of those. Now the other thing that is happening is that I am getting a molecule of ATP. Now I'm going to talk about ATP later when we're talking about energy. But I'm just going to highlight that I'm getting an ATP being produced there. Now this process, just like the link, occurs in the matrix. So we're in the same part of the mitochondrion that we were in before. Now in case you plan on continuing biochemistry and you'd like to know how on earth would I ever remember the names of all of those ridiculous named molecules, this is the mnemonic that I remember learning in my university life. Can intelligent Karen solve some foreign mafia operations? That sentence, the first letter of each word, is the first letter of each of the names of the molecules. Again, I'm not going to ask you the names. That's just in case you're interested, in case you'd like to know. So what I'd like to do is sum up not what happens in each cycle, but what happens in one full cell respiration reaction, because that's usually what we're talking about. And so you'll see underneath, I want to focus on what does one glucose molecule produce, because one glucose makes this cycle happen twice. So for me to give the products, or the things that are important to know that come out of the Krebs cycle, I have to take everything that happened there and multiply it by 2. So since it takes 2 cycles to metabolize one glucose molecule, so each glucose molecule will produce 4 CO2s, 2 ATPs, 6 NADHs, and 2 FADH2s.